Hello students, today we will discuss the last section of chapter 6, which is meiosis and genetic variation. The key concept for this lesson: independent assortment and crossing over during meiosis result in genetic diversity. We've learned about meiosis already in the past lesson, and you know already what crossing over is. So we will just review this one, and I'll discuss a new concept, which is genetic linkage, at the end of this lesson. So the objectives for this lesson: first, describe how sexual reproduction creates unique gene combinations, and explain how crossing over during meiosis increases genetic diversity. One advantage of sexual reproduction is it gives rise to a great deal of genetic variation. So this variation results from independent assortment. Independent assortment states that each number of a pair of allele may be combined randomly with either of another pair of allele. So chromosomes arrange themselves randomly on the equator during meiosis. Okay, there's no specific arrangement; it is random. So after metaphase, wherein they go to the middle of the equator, they will be pulled away from each other during anaphase, and you do not know which one will go to the left or to the right. Okay. So sexual reproduction creates unique combination of genes, and that is because of independent assortment of chromosomes during meiosis and random fertilization of gametes. Okay, so we produce millions of gametes, and you do not know which one from that thousands of or millions of gametes that will fertilize the specific egg. So it's random. Unique phenotypes may give a reproductive advantage to some organism. If you look at this picture, you can see the different phenotypes or different physical characteristics. One has black hair, or another one has blonde hair, with another one has a dark skin or fair skin, or maybe different eye colors. Okay, crossing over during meiosis increases genetic diversity. Let's review again what happens during crossing over. Crossing over is the exchange of chromosome segment between homologous chromosomes. It occurs during prophase 1 of meiosis 1. Okay, you can review meiosis in our past lessons so you can remember what happens during prophase 1 of meiosis 1. And crossing over results in new combinations of genes. Actually, crossing over is also known as gene recombination. Let's review what happens. Crossing over exchanges segments of DNA between homologous chromosomes. So two homologous chromosomes pair up with each other during prophase 1 in meiosis 1. So they pair up, okay? And what is that event in prophase 1 wherein the homologous chromosomes pair up? That is synapsis, right? In this position, some chromatids are very close to each other and segments cross. So they're very close to each other. So their segment, part of this part will cross. Okay, some of these segments break off and reattach to the other homologous chromosomes. So this one is the new combination of the genes. So if you look at this, the 4 chromosomes that would result after the chromosomes go through meiosis, if you look at this, the 4 different chromosomes. One is one full purple, another one has a purple with a green segment, and a green segment with a green chromosome with a purple segment, and one whole green chromosome. So these are the 4 different chromosomes, and they are different from each other. Okay, so that is the reason for genetic diversity. Another concept that I'd like to share is about gene linkage. Okay, I'll have to show you this one. This is one chromosome, and you can see that Chromosomes contain many genes. And in this chromosome, you have gene A and gene B on top, gene C and gene D at the bottom. Gene A and B are not linked. This one, genes are not linked to C and D because they are far apart, okay? They are not linked. Crossing over is likely to occur in the space between B and C. So the crossing over, the exchange of segment will happen in this part, okay, in this part, because B and C are far apart, okay? A and B are referred to as linked because they would likely be inherited together. So A and B is a linked gene. So they can be inherited together because they're close to each other. C and D are referred to as linked. This one, they are linked because they would likely be inherited together. So the separation of the segment could happen between gene B and gene C in this portion. Okay, so this is genetic linkage. So genes, the farther apart two genes are located on a chromosome, the more likely they are to be separated by crossing over. Genes located close together on a chromosome, like this one, tend to be inherited together, which is called genetic linkage. Genetic linkage allows the distance between two genes to be calculated. Okay, so this is the end of the lesson. If you have questions, just message me or email me so I can, if there's a need for me to have a Zoom meeting, aside from our scheduled Zoom meeting, I can do that, but we have to schedule, okay? So this is the end of chapter 6, so I'll schedule a quiz on this section, not chapter 6, section 6. Oh, wait, end of chapter 6, okay? I'll schedule a quiz on a specific topic for you, okay? Bye.